Hello, everyone. Welcome to another ACE Spotlight event. Today, I'm going to host a Spotlight talk with Juan Caicedo. He's going to talk about predicting compound activity from phenotypic profiles and chemical structures. It's going to be a very interesting talk where he's bringing different types of data from genomic data, image data, and chemical structures data in order to facilitate the drug discovery. ACE is a global community of machine learning and AI practitioners and researchers, and we are happy to host topics about different areas of ML and AI. And I usually host topics around the material AI in materials at ACE. So Juan, who is going to talk today about this interesting topic, is a Schmidt Fellow at Broad Institute of MIT and Harvard. He's pioneering the use of deep learning and machine learning methods to analyze microscopy images and high-resolution genetic data. He is exploring reinforcement learning to optimize biological experiments. He collaborates with cell circuits, epigenomic programs, and imaging platforms. He has received his PhD in computer engineering from National University of Columbia. He completed internships at Google Research, Microsoft Research, and Queen Mary University of London as a graduate student. He's a postdoc researcher. He had a postdoctoral research fellowship at University of Illinois Urbana-Champaign. He studied object detection problems in internet scale image collections with deep reinforcement learning. Currently, he's a postdoc at Broad Institute in imaging platform. Without further ado, I'll hand it over to Juan. Please go ahead.
Thank you so much for the kind introduction and also for having me today. It's my great pleasure to present the work that we're doing to predict compound activity using phenotypic profiles and chemical structures. So my name is Juan Caicedo, and I'm a Schmidt Fellow at the Broad Institute. I'm going to be primarily talking about images and how to use images for predicting properties of compounds. And turns out images are a rich source of information, and they can be quantified for all kinds of phenotypes in microscopy. So we at the Broad Institute are interested in processing microscopy images because there is a ton of information present in every image, even a simple image. With modern image analysis, it is possible to measure most phenotypes in many biological models of interest, including complex cultures of cells, or examples that we have here, such as muscle structures, patient biopsies, different types of imaging, including 3D, mass spectrometry, and even time-lapse that can capture changes of images in time. At the Broad Institute, we have developed Cell Profiler, which is an open source software for analyzing and quantifying phenotypes. And I'm also going to discuss today how we use machine learning for extracting information from these images. And this is a specific example of how we can measure properties of images that are of interest for biological analysis. In general, the richness of those images sometimes is simplified too much. For instance, in this case, this is a study for evaluating treatments for AMKL, which is a leukemia type of cancer. And what we expect the drugs to do is to recover the size of the nucleus or the amount of DNA in the cells. And this is one simple feature that we can measure in those images. After segmenting the cells, we can quantify how much DNA is in each of those single cells. And using that information, we can separate compounds or treatments that are actually making a difference from those that are doing nothing, or they are similar to the baseline. So this study was published back in 2012, and it was the first, type of study by a group of researchers that now have a company called Recursion, which is doing a great job at using images as well for developing the platforms for finding compounds using phenotypic information. However, this specific example is focused again in a single feature. And the question in general, when we use microscopy images, is how can we extract more richer features and information from those images? For instance, in this case, we have a population of cells that has been treated with one compound, and we would like to compare this against this other population of cells, which has been treated with another compound. As you can see, there are many visual features that make them different, but we cannot nail it down to a single thing that is measured by biologists. So in this case, we need other strategies to quantify how much variation we observe from the phenotypic reaction of cells to certain compounds with respect to others. And this is what we know as image-based profiling. Basically, in image-based profiling, we have a model, a biological model of interest. In this case, it can be cells. And we run multiple experiments in multi-well plates. Each of those wells has one population of cells, which is treated with a different treatment. And then we fix the cells, we stain the cells, we image those cells to collect this type of visualizations of how the cells are responding to the treatment. And after that, we do image analysis in order to quantify and extract measurements of what is the actual phenotype of the cells. And this has a lot of applications in drug discovery and functional genomics, including identifying drugs and identifying the regulations, the genetic regulators for different types of diseases or models of interest. And in this specific talk today, I'm going to be discussing machine learning for image-based profiling. There are a lot of image processing operations that we need to do in order to transform those images into information that can be used for decision-making. And I'm going to be giving an overview of three major topics. First, machine learning for cell segmentation. Then I'm going to discuss the problem of single-cell representation learning. And finally, I'm going to be describing the results of our newest work, which is predicting compound activity. So let's talk about cell segmentation first. Microscopy imaging is such a versatile technique, which allows us to observe very different cell types in many different details and many different contexts. This flexibility also opens tremendous possibility for studying cell biology, but also poses challenges for algorithms that identify single cells. There are many cell segmentation problems reported in the literature, as many different cell types that are out there in biological labs. And an important goal in cell biology, in cell segmentation problems, is to design algorithms and tools that require minimum configuration. In general, we are still a little bit far away from that goal, but there has been a lot of progress in the computer vision community to design strategies for segmenting objects and identifying different types of objects in images. So given that progress, we also ask the question, what can we do or what should we do in order to have algorithms for cell segmentation that are as robust as face detection algorithms in natural images? Those algorithms can be used in social networks or even in our phones in order to recognize faces without having to do any manual configuration or training any algorithms. They are basically ready to use. But we don't have that in cell biology. So we ask the question, what do we need in order to do that? And back in 2018, we organized the Data Science Bowl in order to invite the wider data science community to help us analyze this problem. This was a challenge that ran for 3 months, and it was the largest data science challenge for social good that has been done at the moment. They proposed a very diverse set of image segmentation algorithms for tackling this problem. And the challenge that we presented was independently of the type of microscopy image, which can be designed by the biologists, can you create an algorithm that can segment those objects accurately? And here are some of the solutions that they presented. In the third place, they had a mask RCNN detection algorithm, which is able to analyze the image, propose some regions of interest, and for each of those regions, they can segment or propose a mask that covers the exact region of interest in the image. This is a popular algorithm in computer vision, so it's good to see that robust algorithms are able to translate into good performance in cellular imaging as well. In the second place, there was a feature pyramid network, which is also very standard in computer vision, and they adapted it to predict the locations of cells with a novel strategy. In this case, they, instead of predicting directly the masks of objects, they wanted to predict distance maps of how far away the edges of the cell are to the center. And with that information, they managed to achieve more accurate results. And finally, in the first place, the winning team put together an ensemble of 32 different neural networks, all of them unit type of networks, which are solid models for doing image segmentation. I think the important message here is that those, in the top 3, we have a diversity of models. So it really doesn't depend on the type of model. It's more about how we train those algorithms in order to exploit the properties of cell segmentation in order to get accurate results. And we are kind of interested in solutions that only have, a lower computational overhead as well. So something like 32 neural networks is probably not going to be used by any biological lab in practice, especially given the differences in performance. They were very minor, and we can get faster segmentation with simpler models. So what can we do with a single model in order to help biologists achieve their research goals? And imagine that you have a biological lab, and you have, during this week, you have to segment a bunch of images in these categories. They represent a quite diverse type of images, and maybe you can have access to some training data in order to create a model for this. So this is the first thing that we can do. We can ask a data scientist to spend some time training algorithms for each of the data types that we have. They display diversity of sizes, colors, and types of objects. And this is kind of the results that we can obtain with 20 hours of work, training unit models specifically for each of the data types separately. Each of the points in this plot represent one image in one of those categories, and the color represents the type of image that we're segmenting. This is the average accuracy that we would get with this strategy, training models specifically for each data type. There is another approach that we can use, which is use existing tools for image segmentation, which require manual configuration as well. So we can ask a novice member of the lab that has no experience with image analysis to go download Cell Profiler and try to segment the images relatively interactively with the tool. This person is going to spend 5 hours of their time in order to segment the same images, and they're going to get pretty much the same accuracy as trained models specifically for each data type. We can also ask an expert image analyst who is going to spend less time because they know exactly what they're doing. And they also will achieve a higher accuracy because they know what parameters need to be adjusted in order to get better segmentation. And finally, if we use the best performing model in the competition, which is based on deep learning segmentation, we don't need any configuration time because the model is trained to analyze any type of image. And we're going to get actually higher accuracy because the model has been trained to tackle multiple types of problems as opposed to the baseline algorithms that we have here, which only benefit of looking a few training examples in each category separately. So this is the type of algorithms that we would like to have for supporting biologists in analyzing their images, something that has no requirements of image processing knowledge and also can give robust performance when segmenting the images. And in fact, there are a couple of existing tools for generic cell segmentation. One of those is CellPose, which can do the cell body or the cytoplasm of cells without any manual configuration from the user, just running the algorithm on top of the microscopy images that they get, and gives a very good segmentation result. Nucleizer is another tool that has been used for segmenting the nucleus specifically. So they are focused on different cellular structures, but they don't require manual configuration. They probably still require more training data in order to, be able to achieve more generic results, but they are focused on doing this type of development, which is the future of cell segmentation. To close the cell segmentation part, I would like to remark a couple of open challenges. Despite the advancements that we have seen in cell segmentation, there are still certain things that can be improved. For instance, compiling a large data set for training models would be of immense value for the community. Many other fields of machine learning have made steady progress thanks to open, annotated data, including the computer vision community and the natural language community. So we will need larger data sets to create better cell segmentation models. And hopefully this requires, collaborations with biologists to include the important types of images in this large data set that we still don't have. And another challenge here for cell segmentation is that even if we collect a very large data set, we are probably not going to have all types of cells represented there. So strategies to quickly learn from new examples with active learning would be also kind of useful in this regard.
Juan, before you go into next section of your talk, could you please elaborate a bit about how big was the data set that you used for training of Cell Profiler?
Yes, thank you so much for that question. The data set that we created had approximately 900 images, and we manually annotated with the help of experts 32,000 individual cells that are represented there. And this data set is publicly available. You can find it either in Kaggle or in our bioimage benchmark site at the Broad Institute, together with the annotations and the partitions that we used for training, validation, and test.
And also these images are 2D or 3D?
We used only 2D images for the challenge. So we don't include, and that's one of the challenge for future research as well. We would like to have, more diversity of images. But in this preliminary data set that we used for proof of concept, to demonstrate that we can segment a diverse set of images, we probably included like 30 different types of experiments. But we know that there are many more that are not represented in this data set. And this is why we are advocating for collecting a larger publicly available set with, more data types, including 3D, including other types of images that we currently don't have. You can find more information about that data set in our website, so I encourage you to go and play with it as well. We would like to learn what you can do with that type of data.
Can anybody use your cell profiler that has been trained and just transfer on their own data set and use a transfer learning type of concepts to be able to segment their own data? Or they need to go through the whole training part again?
That's a good question. I think currently the only tools that we know that are using this type of approach, and just to clarify, Cell Profiler doesn't yet have any of these strategies implemented. Nucleizer would be the most similar one. Actually, Nucleizer uses our data set behind the scenes in order to provide that type of service. But I don't think they share the models at the moment, so you can retrain it. I'm not sure. Maybe you can look at their publication and their website as well in order to, be able to do transfer learning. I believe that transfer learning is also useful for the second challenge that I mentioned here. In order to train quickly new models without having to retrain from scratch, transfer learning would definitely be valuable. And that's a very interesting research going forward, a research direction for going forward.
Is your code available for public use?
The three solutions that I mentioned in the top three solutions have publicly available code that people can use for, shaping their own models or training their own models as well. That, however, requires data science skills, is not readily usable by biologists. And I think that's also another challenge that at least Nucleizer and CellPose are trying to bridge, the gap between having a model that can segment cells and having an actual tool that biologists can use. We hope in the future to see, more tools that are user-friendly. But for now, the code is definitely publicly available. You can, download it. And if you have the skills, you definitely can use it.
And just to be clear, that's three different solutions that you explained about. They are all under the hood of Cell Profiler. Is that correct?
The Cell Profiler doesn't have one of these models implemented yet. This is something that our team at the Broad Institute is working on. But the current version of Cell Profiler doesn't incorporate one of these deep learning models. This was like an open competition that we organized to make a proof of concept, and we are very pleased to see many advances, but we have not been up to speed to incorporate that in our own tool. So we hope to have also contributions. So if you're interested in shaping the code in a better way and maybe contributing it to Cell Profiler, we are also happy to hear from you.
Great, thank you.
Okay, great. So with that, I'm going to move forward to do or to talk about single cell representation learning. So what is this all about? In image-based profiling, our goal is to transform images into information that we can use for making biological decisions. And as you saw just recently, we take the first step of segmenting the images in order to identify single cells. Imagine that in this cartoon, what we have is three different treatments or three different compounds for one specific disease that we're testing. And we want to quantify what is the difference between their shapes and their morphology. So the first step is to segment the cells, go after each of those and find the locations of those cells. And for that, we can use machine learning. The next step, after we have all of the single cells identified in our study, is to measure that morphology of the cells. And that basically results in matrices of features that have single cells in the rows and features in the columns. So we can collect information about what's the morphology of those cells. Maybe the shape of the cell or the size of the nucleus or the distribution of colors, the textures, and many other properties that describe what's the phenotypic state of cells. And with that, we can proceed to aggregate information from the population of cells that were treated with the same compound or treated with the same approach. And that represents the average response of cells to this type of treatment. With those profiles is what we actually use to make downstream statistical analysis or any other type of data science analysis to determine whether the compounds are working or the treatment is effective. So this is the general workflow, the general computational workflow to transform microscopy images into information that can inform researchers to find better treatments or better drugs. So we already saw how segmentation can be solved with machine learning, and we expect this to improve during the next few years. But the next challenge is, how do we measure the morphology of cells in an accurate way? And this is where representation learning can be used. Back a couple years ago, we proposed a strategy to use convolutional neural networks in order to extract features from single cells. Basically, the main idea here is that we are going to train a convolutional neural network to classify the type of treatment that was originally applied to the cells. This is information that we always have because the experiment has been designed in such a way that we know which cells have been treated with which compound. However, we call it weakly supervised learning because we don't always know if the treatment has any real difference with respect to the others. So this is not manually curated annotations. This is what we think the cells, the differences. This is what we think explains the differences between the cells. But in practice, that's the actual hypothesis that we have to test. Remember that our main goal is to aggregate information of multiple single cells and then make a statistical analysis to find differences between treatments. Our goal is not to classify single cells into the types of treatments because we always have that information. We use this as an auxiliary task to learn what type of features the neural network is extracting in order to separate or to find similar treatments downstream. And we are using this approach in order to evaluate whether the biological connections between compounds is useful or not. And in these plots, we have been testing 3 different types of treatments, if you like. This one is a gene overexpression treatment, and this one is compound treatments, these other 2 data sets. And what the plots here display is, imagine that after constructing this type of graph, because what we have here is the similarities between all the compounds, and that's basically a graph that connects compounds to each other, we cut the graph and we only leave 2% of the connections, or maybe 1% of the connections. And this is the percent of the connections that we leave there, the strongest connections in the graph, and we remove all the others that are relatively weak. So what we're measuring here is how many of those top connections in the graph, the strongest and more robust connections, how many of them are actually biologically meaningful. And this is what we measure in the y-axis. The largest, the better. And as we can see, using deep learning features, we can connect the graph in a more meaningful way. This is basically what we read in these plots. This is just a way of generating features that are more expressive and that can separate treatments in a more meaningful way as well. Single cells, this is an additional type of benefit of using images in this case. The original images have single cells, and we have segmented them. So we have features for every single cell in the study. We usually aggregate the information at the compound level in order to do this type of analysis. but we still can go back and look at the differences and similarities between single cells. In this case, what we're trying to convey here is how similar are a couple of profiles when they are aggregated, like the average profile, a couple of average profiles that belong to the same compound or treatment seem to have a lot of things in common. But when you take a single cell and you compare it to the average profile of that population, you start to see less correlation between them. And when you compare a couple of random single cells from the same population, they display a lot of heterogeneity, which is less correlated. So this basically tells us that single cells still have quite a lot of information that we can learn from. And instead of comparing aggregated profiles, we could do single cell analysis, such as in this case, we have a couple of treatments, one that displays a mutant gene, another one which is the wild type gene or the normal version of the gene, and finally, another population that has no gene overexpressed. This is an overexpression experiment. And as you can see, the neural network sees quite a bit of a difference between the three types of populations. More remarkably, the blue population has substructure here, which may explain how the mutant gene is generating the problems that they generate. However, one thing that still needs more research is how to combat batch effects or confounders in the biological signal that we extract from images. In this experiment, each of those points represent a single cell, and the colors in the left are the mutations of genes, whereas the colors in the right are the batches or the experiments that were run. Only 3 experiments to collect multiple, multiple mutations. And as we can see, the major structure of the data corresponds to batches or technical variation, basically. We don't want this to dominate the feature space. We want the major variations in the feature space to be related to biologically meaningful phenotypes. So we need to correct this, and there are several approaches that can be used, such as the gradient reversal layer is one strategy that we tested in this case in order to destroy the structure of the batches and try to emphasize the phenotypes which are of interest here. In this case, this is relatively successful, not as good as we wanted. The colors here should be completely random, but there are limitations in the methods, and this is also an open challenge for future research as well. How can we remove that type of variation from single cell representations so we keep only the biologically meaningful features and we remove other types of variation? So these are some of the open challenges here in representation learning, which includes how can we use domain adaptation in order to do batch effect correction? Domain adaptation is like analyzing images that have been collected in different ways and make them, look like they come from the same domain. This is basically what's happening here in our batch effects problem, that given the differences in microscopic conditions that the images have been acquired, they have a shift in domain, and we should remove that. There is a lot of research in machine learning related to this, including obviously domain adaptation, but also fairness in machine learning. We know that models that are biased end up doing not useful decisions or incorrect decisions sometimes. And that can also happen here. Imagine if this is for real patients or for finding a treatment for a serious disease. We don't want the models to be biased, and this is an important topic of research. We also want to have explainable models with interpretation capabilities because those features that we collect using deep learning specifically usually don't have any explicit meaning. So finding ways of explaining what has been encoded in the representations is also going to be critical to move forward the image-based profiling analysis using deep learning. Okay. With that,
question. Sorry,
yeah,
a question here. Could you please a bit elaborate on the technique that you're using for batch effect correction?
sure. This is called the gradient reversal layer. I don't have kind of a diagram here, but just to give you an idea of what happens, we have a neural network which has maybe I can go back to this diagram in order to help. We have a neural network that has to classify the type of treatment that the cells have been treated with. In order to do the gradient reversal layer, we span another branch here, which is able to classify the type of batch or the domain that the cell comes from. We hope that that specific branch captures the signal that is irrelevant for us. You know, we want to remove that type of variation. So when we train this network that has two branches, the second branch is going to have a gradient that we put in the opposite direction of the optimization problem. So the neural network is going to be optimized for recognizing this and also optimized for destroying the information related to the batches. That's at a high level the idea of the gradient reversal layer. And there are a few papers that you can find if you Google for gradient reversal layer. This has been used in histopathology with success and in domain adaptation as well. So that's kind of the idea.
With this explanation, I wouldn't expect that this batch effect correction can be incorporated in a transfer learning type of thing, because it seems that each experiment or each compound should be trained separately, because the batch effect from one experiment might be different from the another one. Is that a fair interpretation?
Definitely. I think that's an interesting interpretation. I think we need more research in that direction, because even though I agree with your assessment, there is also another assumption here, which is if we encourage the network to ignore that type of variation, hopefully it's going to be able to ignore batch variation in novel batches as well. So imagine that there are, in classical features, there are a bunch of features that we could just remove, and then we get rid of those batch effects. That's not true in reality, but with what the hope here with deep learning is that the neural networks are able to find combinations of features that usually encode the batch effects, and then we can destroy them with this type of approach. I think both types of the argument need to be evaluated with more rigor, I think, because maybe new batches may bring different types of variations that are going to, change our hypothesis here. But on the other hand, maybe if we have enough batches for training, we're going to eliminate the sensitivity of the network to that type of variation in a more generic way. In other words, making it generic to ignore the batch effects would still be possible, I think.
Mm-hmm. Interesting. We have a question in our YouTube channel.
Yeah.
They want to know what you mean by explaining when you talk about highlight the phenotype in the image that explains the disease or effect of compound.
Like in this final open challenge, or more specifically?
No. I think in general, when you're talking about that, the model can highlight the phenotype in the image that explains disease or effect of a compound. What you mean by explain?
Yes, I think that's a great question as well, and I think that's why I list it as an open challenge. We have had discussions with different researchers about what do we mean by interpretation. And this is a not well-defined problem. Sometimes we mean example images, like, give me some example images that represent this specific phenotype. Sometimes we mean regions in a specific image, like, okay, what makes a difference between these two images? Can you point out to the thing that we believe is different? Sometimes it means finding some external type of validation, like correlations with other type of data that confirm the biological hypothesis, and so on and so forth. So explainable models in general are not well defined, and I think that's even a more challenging problem in biology, that it's hard to tell exactly what would be the successful explanation. And I think that requires a lot of collaboration with biologists to be able to, model and design something that makes sense in the context of the project.
A question: when you're showing your single cells that are fed to your network, are you also in your input, will you have a cell that doesn't have any compound introduced or any medication introduced to it, like a control cell that features can be distinguished?
You mean in this one? In the single cell analysis? Yeah. Well, in this specific diagram, this green population is the control population, which means no treatment has been applied to those cells specifically. And the red population was treated with one gene overexpression technique that highlights the activity of one specific gene. I think the gene in this case is BRAF. And in this other case, the population of blue cells have been treated with an overexpression treatment that highlights the activity of this mutation of BRAF. So we want to see the phenotypic differences between those three: the natural state of cells, the normal activity of the gene, and the overexpressed activity of the mutant. Of course, we can compare many more populations in this case, but in the context of this project, comparing the wild type versus the mutant was more meaningful. And for that, we also use the control population as a reference.
Great, thank you.
Okay, cool. Thank you so much for the questions. I think I'm gonna move on to the final version, the final part of the presentation, which is predicting compound activity, which was, in fact, the title of the presentation. So I hope you stick with me in this last part. And, one of the common uses of microscopy images in biology is definitely drug discovery. And at the core of drug discovery is the ability to predict if compounds exhibit a specific activity. In drug discovery projects, institutions may have hundreds of thousands or even millions of chemicals available. And the goal here is to identify which chemicals are useful or potential treatments for the many diseases that still need to be cured. Typically, experts in those diseases design assays or specific probes to test whether the compound is doing the activity that we want. And those include biochemical assays or cell-based assays that are going to turn green, for instance, if the compound is working, or not green if the compound is not working, something like that. There is a lot of chemical engineering behind those assays in order to detect whether the activity is specific to the type of treatment that we want. However, even with automation, it's tremendously expensive to carry out all of the possible assays with all of the possible chemicals for the many diseases that we are interested on. And that's why we have a sparse knowledge about which compounds, which are represented here in the rows, have activity with certain assays or certain probes that are supposed to reveal whether the compound is useful for some diseases or not. And we can't test them all. That's the major problem. So we only know that some compounds are useful for certain assays and some others are not. But for the majority of them, we have no idea. Computational approaches that can help us predict the activity of compounds would be tremendously useful as well. And this was the proposal by a team of researchers back in 2018. They demonstrated, or they wanted to repurpose some existing information in order to predict assay activity in an unrelated type of disease. So for instance, in this case, they used a glucocorticoid receptor assay, which is what the images represent, in order to make predictions in a cancer project. So in principle, they don't have too much relationship, but what this paper demonstrated is that even if they are not directly related, you can reuse existing information in order to make predictions, and then maybe test the compounds that are more promising instead of all of them. So how this approach works? They started using a cell profiler for computing features in the existing images. Remember that this was part of another study, and they wanted to repurpose those for a cancer project. So they transformed those images into image-based profiles using a similar computational workflow as I described before. And then they collected assay activity data for a few assays that they were interested on. Of course, this matrix is quite sparse. They don't have examples of how the compound specifically reacts to, all of the activity that they want to know. But for some of them, they know how, they are, what's the response. So they use these two matrices in order to learn a machine learning model that can complete the matrix, basically. And with that completed matrix, they also estimate the accuracy of those predictions based on cross-validation experiments and so on. So they only keep the assays that are more accurately predictable by the image-based features. And with those predictions, they select the compounds that are more promising for further testing according to those, specific predictions. So that was very useful in their particular study, and they managed to to demonstrate that you can improve hit rates by up to 250 times what you would do if you have to do all the experiments manually in the lab. So this is very promising for computationally predicting which compounds are going to be useful in the drug discovery project. At the same time, another team of researchers were looking at this a similar problem, but instead of using images, they were using chemical structures. So in their work, they basically take the graph of the molecule and they compute features using a deep learning model. This is a message passing type of neural network with a few feed-forward layers. And then they can compute and predict whether that specific molecule is going to be a positive or negative test in some assay of interest. With this type of model, they can scale, to a very large chemical space, even millions or tens of millions of molecules, because looking at the chemical structure doesn't require any experiment in the lab. They only have to look at the molecules, and if the machine learning model recommends that this is a promising chemical, then in that case they proceed to do actual biological experiments, and in that way the model helped them to identify one new antibiotic, which is the first time after 30 years of research that someone is able to predict a new type of antibiotic, which is very powerful and has a lot of interesting properties. So this was a successful case as well. So the question that we're asking here is, can we combine, powerful phenotypic profiling, such as the type offered by imaging, and combine it with the analysis of chemical structures, which is also very powerful for predicting properties and bioactivity of compounds. So this is the approach that we tested here. We take the compounds, and we know the molecule here. So we use ChemProp in order to compute features from the graph of the molecule. And then we use a machine learning model to make predictions in different types of assays. Each of these is a different type of probe or bioactivity of interest. And we can complement this model by making experiments with that compound. Remember that here we only need the molecule, but in practice, if we want to know how the compound actually does something in the cells, we need to do the experiment. And we can make the experiment. We can collect images that tell us what is happening with the cells. And we can create phenotypic profiles using those images. In addition, we can measure the gene expression of the cells that have been treated with this compound and collect gene expression activity in order to complement the decisions of the model. If in the future we want to add another assay here, we don't need to remake the libraries of images or gene expression data. We can repurpose those in the same way as the other researchers demonstrated. All we have to do is to find positive and negative examples of a few compounds in order to make the predictions for the rest. So this is kind of the approach that we wanted to make. And for that, we used the following datasets. There is a few datasets with phenotypic screening and L1000 or gene expression data, which are depicted here in green and blue. Those datasets are publicly available as well, so we basically use them and repurpose those datasets here. And we selected 13,000 training examples from the dataset, and we left out about 3,000 for validation. And that's from the input type of side in the model. For the outputs, we wanted to predict as many assay readouts as possible. And for that, we reached out to researchers at the Broad Institute who were collecting assays of molecules for more than 10 years. So we took the historic of assays that they had, and we found 376 assays, which represent a wide variety of biological activity that has been tested in some of those compounds, not all of them. And in fact, we have 90% sparsity, which means we don't know the activity of or the response of those compounds for the major part of the assays. In total, there are 500,000 results or tests that have been physically run, but the other millions of interactions between compounds and 376 assays is missing. And the type of assays that we found in this list of 376 includes bacterial assays, biochemical assays, yeast, fungal, and other type of cell-based assays, representing a wide variety of biological activity. There are 200 assays that we don't know exactly what type they are because they are confidential. However, we use them anyways in our study to see if we can predict their activity or not. And finally, this is the input model and this is the output model. We used ChemProp to run all of the experiments in this study, which basically takes chemical structures with a message passing type of network. And for the other modalities, we basically create an additional input branch to the feed-forward networks. I want to clarify that the images were pre-processed using classical features instead of deep learning features. So there is room for improvement in the results that I'm going to share. And also, similarly for the gene expression data, it's concatenated in another branch to the feed-forward network. So in practice, this network is going to have 3 inputs. One which does deep learning on the chemical structures, and the other ones that receive pre-computed features for the compounds using imaging or gene expression. So here are the results. The performance of individual modalities. This is the number of assays that we wanted to predict, and this is the accuracy offered by each of the models. Cell painting represents morphology extracted from images. L1000 is gene expression in blue, and the chemical structure is what we can, just the graph of the molecule. And the higher the number of assays, the better the performance, of course, which means that if you are comfortable with accuracy level of 0.7, you're going to be able to predict 194 of the assays that we have in the list using only imaging. And you're going to be able to predict 174 with chemical structures and then 112 with gene expression. We found that all of the 3 modalities complement each other, and they seem to have different, they can predict different type of activity because only 3 of those assays that can be predicted by any of the data types are in common among all of them. So this is, interesting for future research. In this case, we are testing the individual modalities as opposed to combining them because we wanted to know how each modality can make the job without the others. We also analyzed the performance of combinations of modalities, and we basically used basic data fusion. As I mentioned before, in the neural network, we added branches for the imaging features and the gene expression features. And we found some challenges in making this work. Seems like there is still room for improvement as well for being able to assign better importance to different types of data modalities. And we see that the combination of chemical structures and morphology is the one that has more success in general, which is this one. And but if you can, mix them all as well, we're going to lose the ability to predict some of those assays. We found that, combining the assays, we can predict up to 71 assays at this level of accuracy. So this is the very high level of accuracy, 71 assays. And if we do retrospective analysis, it's possible that using just the, the single best is going to give us 92 assays that can be predicted with chemical structures and morphology. And I just want to summarize all those numbers to say, when you take all of them, you can predict up to 100 assays with very high accuracy, which is more than 90%, using all of the 3 modalities simultaneously. We still believe that there is better ways of combining the data using other, types of neural network architectures that, for instance, use attention in order to decide which part of the data is useful, but that's not what we used in this study yet. We wanted to test whether the different modalities are useful for predicting compound activity. And compared to the hit rates that biologists observe in the lab, so, imagine that they are testing 1,000 compounds, and if they find one of them which is useful, they're going to have a very low hit rate. If they have 10 or 100 compounds, they're going to have higher hit rate. So this is what the x-axis means. And what we measure here is how many times you can improve that hit rate that physically that is physically observed in the lab when you do the manual experiments versus using a machine learning model that can help you predict. And in most of the cases, the machine learning model is able to improve up to the theoretical limit. And when your compounds are hard to find because they are not as common, in those cases, the amount of improvement that you get is obviously higher because the machine learning model can find a subset that is more useful for making the predictions. We also found that the performance of this model doesn't quite correlate well with the size of the training set. There is usually the idea that with more data, you can predict better. And I think that's still the case, but that's not what the data tell us here. This is the number of examples in the x-axis, and we have more data here, and this is the accuracy of the models in the y-axis. So as you can see, with a few examples here in the x-axis, we still can do a pretty good job for some of the assays. Each point here represents one assay in the dataset. So there are a few that can be accurately predicted with low number of training examples. And the opposite is also true. With a lot of examples, we sometimes cannot reach high accuracy. So we believe this depends on the specific type of biology behind the scenes. So yeah, so this is something to do more research about as well. Okay, so in summary, in this assay prediction study that we did, we found that morphological profiles are the best individual modality for assay prediction. If you add morphological profiles to chemical structures, you can improve the prediction of compound activity. And the models can predict quite a diverse set of assays, and the modalities that we used are actually complementary to each other. They have different strengths. And finally, about a third of the assays in our study can be predicted computationally, which means that, instead of doing physical experiments only in a pharmaceutical lab or something like that, we can do data science and we can prioritize the assays using artificial intelligence. I think with that, I basically want to wrap up saying that image-based profiling is maybe one of the technologies that is going to push forward biological discovery and drug discovery. And I like to make this analogy with diamonds, from single cells and raw images. We need tools to transform that information into something that is going to be not just beautiful, but useful for doing biological discovery. So those tools that transform raw diamonds into, jewelry is the analogy that we need here. We need tools in order to transform that into useful information for making biology. And with that, I want to say thanks to Tim, Kevin, Shantanu, Anne, and all of the other people and friends, colleagues, and collaborators that we had in order to complete so many of those studies. And thank you all for paying attention to this talk.
Thank you, Juan. Really interesting talk. I enjoyed especially how these different models complement each other and how accuracy can be improved with combining different resources of data. We have a few questions in our YouTube channel, which I would like to relay them to you. One is, if you compare experimental images to a neutral cluster center, can you see differences using a bounded or a range metric like cosine distance? Would you consider that a form of interpretability?
Okay. So I guess this is back to the previous section. And can you repeat the question, please?
It's saying that with comparing experimental images to neutral cluster centers, can we and seeing the differences using a cosine distance, can we consider it as a form of interpretability of the model that you said we are lacking?
Absolutely. Yeah, I think the distances between, known treatments, and in this case, the suggestion is, for instance, neutral or control populations of cells. And the other treatments are definitely meaningful. And this is one of the things that we routinely use in order to try to check, it's like a quality assessment that we make, whether we are really capturing something biologically meaningful or not. So I completely agree that beyond the features, looking at the connections between the data points that we have and whether the graph structure is organized in a meaningful way. Because even though this is kind of similar to the assays, we don't know everything about biology, but we have some data points very well annotated. And if they are, in good proportions in the feature space, that is helping us interpret the entire set in a meaningful way. So I think that's a great idea. Oh, sorry, I can't hear you. I think you're muted.
Sorry. Yeah, I'm muted. Another question that we have is like all the cells in the microscopy images, average the cells in the field of view. Cell paint profile in the field of view.
Yes, and that is going back to how we build profiles here. This is definitely one of the strategies for aggregation, just computing the average of the population of cells, and that's going to give you one baseline here. There are some other methods in which instead of the average, you can aggregate information in other ways. But the baseline for this type of studies is what happens if we only compute the mean response of the population in a single vector. So that's correct.
Mm-hmm. Another question is related to, I think, the last part of your presentation regarding the more data that you introduced to your model. It's saying that can it be the quality of data that is causing that issue that you won't get improvement in adding more data?
Yes, that's a great question. That is possible, a possible explanation as well. One of the reasons is, as I mentioned, we didn't use deep learning for transforming the imaging data or the gene expression data. We used classical features aggregated with classical algorithms. So maybe we can improve the signal that we obtain from those two, and maybe that's going to change the landscape here. So that's one thing. The other thing is also the quality of the data in general, not necessarily the way we process it, but the quality of the data. That's also, that can be also a concern in practice. And I think this is the type of question that biologists always ask. Are we capturing in those images the real biological activity, or maybe there is some noise that is confounding our signal here? So that's a genuine question, I think, and we did some quality checks on the data that we used. The original data set had 30,000 compounds, 30,000, but we only kept, if you do the math here, about 17,000 compounds. So we actually rejected a bunch of compounds that were not useful for making this study, in part because we didn't have any relationships with some of the assays here, but also because maybe the gene expression data is not of good quality, or the cell painting data is not of good quality, and then we had to remove some of those from the study. We believe that we kept good enough quality of data, but the question behind, I think, is definitely a good question to keep in mind.
Thank you so much. We really enjoyed your talk, and thank you again for joining us for this event.
Great.
With that, I would like to thank you, and thank you our audiences who joined us online with their great questions. That's okay. Yeah. I wasn't sure that I can move.